after pollination process, either by natural pollination or by artificial pollination process. So, the final step which leads to the formation of seeds is the fertilization process. So, fertilization means the fusion of male and female gametes. So, here what is the specificity or what is the significance of double fertilization, where we can observe, in which type of plants we can observe double fertilization means this is a special mechanism that is observed in only in the angiosperms. No other plant species will exhibit double fertilization. So, here fertilization is taking place twice. So, that is why we call it as double fertilization, in which male gamete will fuse with female gamete to produce zygote. As we know that male gamete is haploid, female gamete is also haploid, resulting into zygote which is diploid in condition. And this is a first fusion where we call it as a first fertilization or syngamy process. We call this as syngamy process. Whereas here the second male gamete is present, because the the generative nucleus of the male gametophyte will produce two male gametes. The first male gamete is fusing with female gamete, whereas the second male gamete, which is haploid in condition, will fuse with the secondary nucleus of the female gametophyte. The secondary nucleus is diploid in condition, and it is resulting into a triploid structure called as primary endosperm nucleus, or we can also call this a primary endosperm cell. Here one set of chromosomes and two set of chromosomes, that means total of 3 sets of chromosomes are fused. So, we call it as triple fusion, resulting into a triploid structure, that is primary endosperm nucleus. So, here 2 times of fusion is taking place, that is why we call it as a double fertilization process, which is unique to the angiospermic plants. So, let us see how this mechanism is taking place in the angiosperms. Because of pollen pistil interaction, the right type of pollen grain is identified by the stigma, so that it will secrete some chemicals and allow the pollen grain to germinate. This pollen grain, when it is germinating, the intine wall will be converted into a small tube-like structure which will pierce to the stigma and style, and finally it will enter into the ovary, from there into the ovule. In the meantime, when the pollen grain is passing, the generative nucleus will undergo mitosis to produce 2 male gametes. So, here all this process starting with the receiving of pollen grains and till the pollen grains producing pollen tube and the male gamete production, till the entry of pollen tube into the ovary and ovule, all that will be the part of pollen pistil interaction. So, this is a dynamic process which is taking place in the plants. In this process, first one is entry of pollen tube into ovule. The pollen tube enters into ovule by 3 mechanisms. The first one is porogamy. Consider this is the ovary. This is embryo sac. So, this is chalaza, micropyle, funicle like. So, the pollen tube enters into the ovule. This is a pollen tube. The pollen tube enters into ovule through micropyle directly. So, this mechanism is called as porogamy. This is most common type, can be seen in examples like hibiscus and otilia. The second one is chalazogamy. In this, the pollen tube enters into ovule through chalaza. So this is a chalaza. The pollen tube will enters into ovule through chalaza, and after entering into chalaza, once again it will move into the integument region, where finally it will reach the micropyle like this. So as the pollen tube is entering into the ovule only through chalaza, so that's why we call it a chalazogamy. This mechanism was first discovered by tube scientists in Casuarina plant. And third one is mesogamy. In mesogamy, the pollen tube enters into ovule either through integuments or through the integuments, or through the funicle like this. This type of mechanism is called as a mesogamy. This can be seen in cucurbita species. So likewise, the pollen tube enters into ovule either of these three mechanisms, either by porogamy, chalazogamy, or by mesogamy process. But in all of them, one common character if you could observe, finally the pollen tube is coming to micropyle only. In all of them, finally the pollen tube is coming to micropyle. From there, the pollen tube will enter into the embryo sac. Entry of pollen tube into embryo sac. Here, finally, as I said that the pollen tube is coming to the micropyle, the pollen tube will enter into embryo sac only through micropyle. Only through the micropyle. Once it is coming to the edge of this embryo sac, so as we had discussed that the filiform apparatus which is present in the synergids will direct the movement of pollen tube into the embryo sac. So here the next one is entry. In the entry process, what happens, the pollen tube will enters into the embryo sac either piercing through one of the synergid. So when it is piercing through one of the synergid, that synergid cell will get degenerated or destroyed. The other mechanism is the pollen tube will enters into embryo sac between the egg cell and the synergid cell. A small gap like structure will be there. Through that the pollen tube will enters into the embryo sac. Once the pollen tube enters into the embryo sac, so the tip of the pollen tube may burst, or a small perforation may be formed at the tip of the pollen tube, or the pollen tube tip will get degenerated. Because of either of these three reasons, the small perforation will be formed at the tip of the pollen tube, and the two male gametes which are present in the pollen tube are released into the embryo sac. Once the release of male gametes into embryo sac occurs, one of the male gamete, so this is one egg cell we can consider, and this is a synergid cell. So why I am showing only one synergid cell means because one of the synergid had got already been destroyed. So this is a pollen tube. So this is the egg cell. In this is the nucleus of the egg cell, and here we can see the central cell, or we can call it as a secondary nucleus. When the two male gametes are released, these are the two male gametes, one of the male gamete will move towards the egg cell, and other male gamete will move towards the secondary nucleus, and the one which is moving towards the egg cell, so that will get fused with the nucleus of the egg cell. So we can show like this, both of them are getting fused here, and one other will be moving like this. So here secondary nucleus, this is a secondary nucleus, and This is a second male gamete. So this, when these two are, that is female gamete, the nucleus of the egg cell, and the male gamete are fused, so that will be resulting into zygote. Whereas this will get fused with this, the second male gamete will fused with the second nucleus, so that will be resulting into an triploid structure that is called as primary endosperm nucleus, which is triploid in condition. So already we had discussed that zygote is formed as a result of syngamy, the first fertilization, whereas primary endosperm nucleus is formed as a result of triple fusion or the second fertilization process. And here syngamy was first discovered by Strasburger scientist, whereas triple fusion, the result of triple fusion is primary endosperm nucleus. Triple fusion was identified by Navashin, first discovered by Navashin scientist in Fritillaria and Lilium plants. Fritillaria and Lilium plants. So later Navashin had come to know that it is not only been observed in Fritillaria and Lilium, it is a common feature present in majority of the angiospermic plants. So likewise the two types of fertilization is taking place, so we call it as a double fertilization process in angiosperms. So during double fertilization, how many genomes of male gametophyte and how many genomes of female gametophyte are participating? So let's take the male gametophyte side. As we know that male gametophyte contains one vegetative nucleus or vegetative cell and two male gametes. So this is haploid, that is which contains one genome. This is also haploid. So as there are two male gametes, two genomes are present. If we take the side of female gametophyte, the female gametophyte or embryo sac which contains one egg cell which is haploid, two synergids, these are also haploid, one secondary nucleus, this is diploid, and three antipodals, each of them are haploid. So here one male gamete, that is one genome of male gametophyte, and one egg of female gametophyte are participating in syngamy. So syngamy, for the process of syngamy, male gametophyte side and female gametophyte side, how many genomes are participating? One genome of male and one genome of female are participating, whereas for triple fusion, the second male gamete, that is one genome of male gametophyte and two genomes of female gametophyte. Two genomes means the diploid nucleus of the secondary nucleus is participating here. So the total here two genomes and here three genomes. That means for double fertilization a total of 5 genomes are participating. In double fertilization a total of 5 genomes are participating, and how many genomes of male and how many genomes of female? So 2 genomes of male gametophyte and 3 genomes of female gametophyte are participating in the double fertilization process. And how many genomes are degenerated or how many structures are degenerated after fertilization process? The 2 synergids will be degenerated and the 3 antipodals are also get degenerated. So total 5 genomes, 5 genomes are degenerated, which are not participating in the fertilization process. So this is about the number of genomes and the cells which are participating in double fertilization and degenerated, not participating in the fertilization process.